I've been told to make it a very general talk, so it might maybe bring out some collaborations rather than focusing on particular projects we're working right now. So, okay. So you're not going to hear AI that much. You're not going to hear neural networks. You're not going to hear anything deep or deeper. It's going to be a very micro-scale talk. So with past probably 2 decades or so, we've been very fortunate to have this advanced neuroimaging modalities to monitor the function and the structure of the brain and other body organs non-invasively in vivo. And this actually allows us to better understand the diseases, the pathophysiology, the progression of the disease, and how the body is reacting to the treatment or the prevention. So from neuroimaging perspective, we have various modalities that that allows us to quantify structural properties of the brain. We have various functional neuroimaging modalities where we can look at like the synaptic function, metabolism, blood flow, different aspects of the physiology of the brain. And most recently, we've been working on molecular neuroimaging approaches where particular proteins can be targeted using particularly either positron emission tomography or SPECT imaging that actually allows us to study neuropathologies in vivo rather than waiting until autopsy. So with all those research-related advancements, yet we're still seeing a very limited use of neuroimaging in degenerative diseases or psychiatric disorders, both in clinical practice as well as in clinical trials. So when we talk about clinical practice, that's basically using imaging to do diagnosis or prognosis, or in clinical trials, using imaging as biomarker for primary outcome measures, or using imaging to do participant screening. And this is very different than what we see in, for instance, cancer. Imaging plays a huge role both in clinical practice as well as in clinical trials if you think about cancer, but that's been very, very limited for neuroimaging. So my center and my primary focus is really looking at biomarkers and trying to answer some key questions from clinical practice and clinical trial perspective. So why do we need biomarkers? Because we think that will allow us to do diagnosis with higher sensitivity and specificity. And we also want to have biomarkers to accurately or with more precision monitor the disease progression. And also to use biomarkers to measure the treatment effect or the efficacy of the treatment or predict the efficacy of the treatment. So when we think about the current practice in neurodegenerative diseases or even psychiatric disorders, what we're relying on is more clinical diagnosis or what we call clinical endpoints. These are basically based on cognitive or clinical assessments. And we use terms like having dementia or having Parkinsonian syndromes. And some of them are actually used across different diseases. So we have something called clinical dementia rating, and we keep seeing that in Alzheimer's disease. We keep seeing that in frontotemporal dementia. We keep seeing that in vascular dementia. So it's been used in cross domains of different diseases, which actually shows how non-specific it is. It might be very sensitive to pick the dementia state or the disease, but when we're at a stage where it's important to differentiate different diseases or different disease status, these terms, this clinical diagnosis or clinical endpoints, becomes very non-specific. And also it lacks the state versus the stage differentiation. So like identifying someone with mild cognitive impairment is important, but also identifying what is the stage of that individual in terms of the progression, in terms of within how many years or when, if or when that participant, that individual will develop full-blown dementia. So knowing state is one thing, but knowing the stage of the syndrome or the disease is also very important from clinical and clinical practices and also for clinical trials, particularly for clinical trials. So the hope is with the advanced imaging techniques, particularly molecularly specific biomarkers, we might be able to have improved diagnostic accuracy, and we might be able to track the disease progression objectively rather than relying on those subjective clinical or cognitive assessments or the questionnaires. And also this will allow us to do quicker screening for clinical trials. One of the big hurdles for clinical trials is they have to screen lots of individuals for their trials. That's like thousands of individuals, and most of the time a big percentage of those trial participants actually don't have the pathology that the drug is targeting. So it's, that actually leads them to screen more and more and enroll more participants to show the efficacy of their treatment. So this is the motivation for us to work on biomarkers, and with big data coming, even for neuroimaging domain, there's a huge potential for artificial intelligence, data science to pick the proper biomarkers to answer all these questions. So starting with the biomarkers, what are we focusing on? Since my research is really practical research, and most of the questions we're targeting are clinically motivated rather than doing pure mathematical science. We always need to check with what the agencies want from us. So biomarker, the word biomarker is actually very old, and NIH and FDA actually recognize that word. And at some point, NIH actually decided to formally define what biomarker means, because people have been using it, and they decided, let's put a formal description, because it goes all the way back to 1980 when some of the cancer and heart disease research talk about biomarkers for the disease. So basically, it's an objective measure or evaluation as an indicator of the normal biological process, pathogenic processes, or pharmacological response to a therapeutic intervention. It's a very simple definition, basically, but the key is being objective, something that can be reproducible, something that can be translated to different cohorts or different disease stages. It's not going to be one particular domain, one particular study showing that as an important biomarker. It needs to be replicated again and again. If we look at Alzheimer's disease, that's one of the hottest topics since there's lots of funding, and we're getting a huge elderly population that might develop Alzheimer's disease pretty soon. There's lots of effort to better understand Alzheimer's disease, prevent or cure Alzheimer's disease. So when we look at the Alzheimer's disease literature, the most reproduced, replicated biomarker is actually hippocampal volume. And it's something we extract using structural magnetic resonance imaging. And it's been shown that the greater hippocampal volume loss in early Alzheimer's disease, or even in the risk gene for Alzheimer's disease, the ones who carry that risk gene, we see greater hippocampal volume. As disease progresses, the rate of hippocampal volume actually increases. So it really reflects the disease, the dynamic of the disease, and it actually allows us to better understand the natural history of the disease. But yet again, it's not a very specific biomarker for Alzheimer's disease, because if you look at people with major depressive disorders, they also have hippocampal atrophy, and sometimes it's the same order as what we see in Alzheimer's disease. Or if you look at frontotemporal dementia, vascular dementia, they do produce hippocampal atrophy. If you have epilepsy, certain epilepsy groups, they also have hippocampal atrophy. So it's very sensitive to understand the disease progression in Alzheimer's disease, but it's not a very specific biomarker. The game changer for Alzheimer's disease was actually the development of the amyloid imaging. This is one of the neuropathologies that has been linked to Alzheimer's disease. There are two pathologies for sure diagnosis of Alzheimer's at autopsy. One is beta amyloid protein accumulation. The other one is tau accumulation. And once we were able to do in vivo screening for amyloid pathology accumulation, what we observed is what we call clinically Alzheimer's disease patients. About 25 to 27% of those participants, those AD patients, actually don't have AD pathology. At the same time, when we look at what we call cognitively normal, these are clinically normal individuals. They don't have any symptoms. They don't have any disease at all. But when we look at their brain and the protein accumulation, about 1/3 of those cognitively normal, healthy individuals actually harbor Alzheimer's pathology without any clinical symptoms. And this actually was a big game changer in Alzheimer's disease, as well as the way people look at Alzheimer's disease diagnosis clinically, what we call the amyloid phenotyping. It's not just a clinical diagnosis, it's actually phenotyping based off a biological marker using imaging. So what is the effects of using amyloid status? If you know, for instance, mild cognitive impairment is one risk factor for dementia. If you look at MCI cases without any amyloid accumulation in their brain using imaging, it's a very small percentage of those individuals that convert to Alzheimer's disease. But if they do have amyloid in their brain, about 1/4 of the MCI participants convert to dementia within 2 to 3 years, which is a very short period of time. So it's a huge impact knowing that having that biomarker and knowing what it means for the disease progression. Also, if individuals with amyloid in their brain, regardless of their cognitive state, this is basically an individual with a normal cognition or an individual with MCI or AD diagnosis, if they have significant amyloid accumulation, those are the ones that shows the highest rates of cognitive decline. Again, it's very important to understand the stage of the disease, where it goes, and how fast it's progressing. So looking at longitudinal studies in normals, it's a very crowded graph, but there are two things I want to highlight. The first one is there is 2 different studies here. These are 2 different cohorts, actually done in 2 different continents. One is in the United States, Alzheimer's Disease Neuroimaging Initiative. The other one is the Australian Imaging Initiative, again, for Alzheimer's disease. So this is totally 2 different etiology and population. Looking at amyloid positive and amyloid negatives within this cohorts, we definitely see this and looking at different clinical and cognitive measures. This is mini-mental state exam, clinical dementia rating, and logical memory processing. There's a clear separation between amyloid positive and negatives, but this separation is independent of the cohort. It's a very replicated, reproduced finding. Again, coming back to what biomarker is supposed to do. Detection of, since then, it's been very clear that detection of brain amyloid can be used as a risk factor, also can be used to predict future decline to Alzheimer's disease at different disease stages. And combining this biomarker with other biomarkers actually improves the power, our prediction power. So this is an example where genetic information, in this case, it's ApoE4, and other biomarkers that is targeting the tau pathology in the brain, as well as the hippocampus, which is the most replicated and well-established biomarker for AD. Just combining all these different biomarkers together, this was where machine learning came, actually. We can actually increase our power to predict their disease progression. So coming back to the clinical trials, this is data from a real phase 3 clinical trial that's done a while ago, about 10 years ago, where we didn't have amyloid imaging available, actually. And this is a post-analysis of the clinical trial data, just looking at their amyloid, it's a failed AD trial, by the way. It didn't work. But when we went back and analyzed their data, one thing we noticed is they were recruiting mild Alzheimer's disease patients from clinical perspective. But from a biology perspective, only 73% of their AD patients actually had AD pathology. And the treatment was really targeting the amyloid accumulation in the brain. So the goal was removing amyloid accumulation and hoping that will improve their clinical and cognitive performance. But when you have about 27% of your participants not having the pathology you're targeting, of course you're not going to show any improvement because they don't have the pathology. And it's like you have a medication for migraine and you're giving that to someone who doesn't have migraine, and you're not going to get any change in their mood or their symptoms. It was the same story. This is when they only actually have the amyloid imaging done on a very small percentage of their cohort. They were only able to afford that for about 1/3 of their cohort. So the rest of the cohort they studied in this trial, they didn't even know what was the amyloid status of that cohort. So in this collaboration with this clinical trial, we propose we can use imaging data, their MR imaging data, to impute their pathology. So again, this is another area where you can use machine learning. If you have a training data where you know the pathology, you have some other imaging data, and you learn from that, and then you use that to impute potential pathology on those cases you don't have actual biomarkers. And this is what we did. We were able to find key imaging markers that might be a good predictor for their amyloid pathology without doing any amyloid imaging or without doing any invasive approaches to get their amyloid status. And we also showed that if you have not just structural imaging, but also have other functional imaging included in this prediction model. In this case, it was a blood flow versus atrophy. You can actually increase your power to predict, your accuracy to predict their amyloid positivity versus amyloid negativity. And this was a project we did with this clinical trial, and they went back and looked at they reanalyzed their data, basically taking that 2/3 of their participants who never had amyloid phenotyping done using any biomarkers, but just using their structural MR imaging to impute their amyloid positivity. And then we analyzed their cognitive and clinical progression throughout the clinical trial. So it's another very crowded slide, but the reds are basically amyloid negative ones, either based on actual amyloid imaging or based on imputation using MR imaging. And the blue is amyloid positive individuals, again, either based on real imaging, amyloid imaging data, or based on structural imaging, we just predict their amyloid status being amyloid positive. The key finding from this retrospective analysis of their clinical trial is they have this huge cohort, didn't even decline at all because they were amyloid negative from clinical and cognitive assessment perspective. And this was what was diluting their clinical trial findings, and statistically it was a failed trial at the end. Yes. Yeah. This was about 7 years ago, and since then, if you look at particularly Alzheimer's related clinical trials, they actually use amyloid imaging or CSF biomarkers to do amyloid phenotyping when they recruit participants. To maximize their clinical trial statistical power. And this is something that's been acknowledged and recognized by FDA at this point. They are really allowing use of imaging markers to do screening to really facilitate successful trials. Because still we don't have any successful Alzheimer's disease trial in our agenda. So they really want, they're encouraging use of biomarkers, particularly amyloid screening, to identify the really relevant participants for the clinical trials. And a couple months ago, they actually started drafting a new guideline, FDA, again emphasizing the importance of biomarkers for clinical trials. And this time, they actually went one step further. They're also emphasizing the importance of biomarkers, validated biomarkers, to better understand the stage of the disease. It's not just finding amyloid-positive participants to make sure they do have the pathology and the disease. They're also encouraging using biomarkers to see which one of those are actually declining fast, because those are the ones you want to enroll in your trial to increase your statistical power. So it's not cognitive decline, the rate of clinical and cognitive decline, that is the key for clinical trials as well as clinical practice. If someone is very stable, that's a different story, but if someone is progressing fast, that's, your treatment will be different, and the priority for clinical trials will be different as well. So going back to the imaging research we're doing, we've been looking at different imaging modalities to better understand what actually predicts the cognitive decline at different disease stages. So this is a study where we looked at amyloid accumulation and the rates of atrophy, structural change, in a very blinded way. It's not like one location atrophy versus one location amyloid. This is really looking at the entire imaging data, every voxel simultaneously, all the modalities simultaneously, to identify co-varying changes, both structural and pathologically, and asking the question, what are those, and to what extent those co-varying changes in the brain predicts cognitive decline. So in that study, as expected, there is frontal and parietal amyloid accumulation that is highly associated with increased rates of atrophy in the medial temporal lobe, which is very important for memory, which is the key for Alzheimer's disease development. And it was this atrophy, which is a marker of neurodegeneration, that was the best predictor of greater cognitive and clinical decline. It again fits what we know from autopsy studies, but this is a way to really replicate and show in vivo and make it more clinically relevant in real life. And another thing I want to emphasize here is Alzheimer's disease is associated with amyloid pathology. You have to have amyloid pathology. That's the definition of the disease from autopsy studies. And that's how Alzheimer's defined the disease at the beginning. But even in the autopsy studies, it's been shown again and again. It's not actually it's not the change that correlates with the disease state or the cognitive decline. It is, you have to have amyloid, but something else is actually more important important for cognitive and clinical decline. And this is what we see again and again in imaging data as well. This is one example. Amyloid accumulation is relevant to atrophy, which is a marker of neurodegeneration, but it was this neurodegeneration that was associated with cognitive decline, not the amyloid levels. So this is where the tau imaging actually becomes very important. Tau imaging is very new. It's been probably 4 or 5 years at this point, and the last 2 years we've been very active in tau imaging. That's the other pathology you need to have in Alzheimer's disease. And again, the whole idea is even when we look at normals, about 1/3 of us actually have amyloids in our brains, but we never show any cognitive decline. So that is, at some point, having that amyloid needs to facilitate or trigger some other mechanism. That's, from autopsy studies, we think that is the tau accumulation, which leads to synaptic dysfunction and neurodegeneration. That ultimately leads to what we see in clinic, those clinical symptoms. So this was another study where we looked at multiple modalities, whole brain analysis, and to understand the rate of amyloid accumulation using imaging data and how that relates to the tau burden in different brain regions. Again, it's a whole brain analysis. We're looking for covariant associations, clouds of brain regions having greater amyloid accumulation and how that relates to their tau accumulation. Not surprising, but it's, again, some of the parietal regions, if there is an increased amyloid accumulation, it is associated with greater tau burden in regions that shows up in later disease stages. And again, when we ask the question how these relate to their cognition, it was tau burden being associated with greater cognitive or clinical decline rather than amyloid. So amyloid is important, but it's not the key player if you're focusing on their clinical or cognitive changes. I'm almost done. These are kind of the domain knowledge we've been collecting, building over the years, past 2 decades. And this is where I'm going to start talking about machine learning. So we know atrophy is important, amyloid and tau is important, and we know it's not really local, it's the whole brain and how these different pathologies and brain changes are interacting that defines the clinical and the cognitive outcomes. So that there's this network interaction idea, and the question is, using this domain knowledge, can we actually predict who's going to have fast cognitive decline or fast clinical decline that can be used either in clinical trial, participant screening, or in clinical practice in terms of doing prognosis on your patients? So one way to do that is using machine learning, and let data tells us if this is something possible. So this was one example where we used the graph structure of the brain, the connections on the brain, and use different modalities to quantify amyloid, tau, atrophy in different brain regions, and their relationships in this graph to predict who's going to decline fast or slow in different cognitive domains. In this case, it was memory or executive function, and this is where we use convolutional neural networks defined on graphs, brain graphs. And it's very early stages, but even with this very simple model, using what we know about the disease pathology, we are able to predict who's going to decline fast or slow up to 92% in a validated cohort. It's from positive predictive value point of view, it's a big improvement for clinical trials as well as clinical practices. So just to wrap up, the big picture for us, for our research team, is there's no treatment, but the goal right now, the focus right now is really slow the progression. And in order to do that, you need to predict who's progressing fast. And to accomplish this, we need biomarkers to identify who has a disease, the real disease in terms of the pathological definition, as well as who will progress fast. So that those are the 2 key questions we've been focusing on. And these needs to be clinical biomarkers. We cannot have something extra fancy. It needs to be very practical that can be done at any clinical center or any hospital, like simple structural MR imaging, nothing amyloid PET or tau PET. Those are very fancy. That's just a summary of what I've been telling. It's a big team effort. There's lots of funding on this, both from NIH as well as from pharmaceutical companies and foundations. I'm just going to do a couple more advertisements. We actually are looking for 2 postdocs, fully funded postdocs, with some data science and big data background to work on similar questions. In addition, we're actually searching for faculty level applicants at the VA, Veteran Affairs Medical Center. One position is focusing on the big data problem. And the key for this position is we want it to be clinically translational. The application needs to have a clinical translation. It's going to be an imaging position, but the questions will be all focused on clinical applications. And the position actually has about 50% hard money coming attached to it, so it's a 50-50 VA UCSF position, if someone is interested in.